Hi, this video will explain about host cell and modifying enzyme, another two tools that we use in genetic recombinant DNA technology. What is a host cell? Host cell is a cell that receives the recombinant DNA for cloning purposes. Host cell in Malay we call it sel perumah. So sel perumah host cells is a very general term used in many fields. But for cloning, a host cell is the cell that receives the recombinant DNA. How can a cell become a good host? How can a cell become a good host? So a host cell must able to receive or accept recombinant DNA. The process of them to showing that they are able to accept the recombinant DNA is called transformation, right? When we insert the recombinant plasmid, this is the recombinant plasmid. During the insertion, when we insert the recombinant plasmid into the host cell, that process is called transformation. The definition is a process by which recombinant DNA is transferred into host cell. The second characteristic is host cell must be able to maintain the recombinant DNA from one generation to another. If they receive the recombinant plasmid but they cannot express or they cannot maintain, the next generation does not have that particular characteristic, then the cell cannot be a host cell. Next, the characteristic is the host cell should be able to amplify the gene product. Amplify here is has a similar meaning with multiply. So they need to be able to amplify the gene product from the recombinant DNA. So when they do cell division, a bacteria reproduce through binary fission, right? So when they reproduce or they undergo cell division, the recombinant plasmid needs to be replicated too. If not, then they cannot maintain the recombinant DNA. So the daughter cell will not have the similar recombinant plasmid like the parents have. And the next characteristic is for them to be able to express the gene. If they only carry the gene but they cannot express the gene, meaning the gene must be able to be transcribed and then translated into a polypeptide chain. But if that process cannot be done for that particular gene, then the host cell is useless. Okay. The next important tool is modifying enzyme. So what is modifying enzyme? It is an enzyme that involved in degradation or synthesis and also alteration of the nucleic acid. Alright. So modifying enzyme that commonly used is DNA ligase. So do you still remember DNA ligase during in replication of DNA ligase joins the Okazaki fragment. Okay. So in this process of gene cloning, uh, it, DNA ligase does the same function. They join the furin DNA fragments that contains the gene of interest and the bacterial plasmid. Before, we have cut open the plasmid, right? So, the plasmid, and then when we combine the gene of interest with the plasmid, and then we put in DNA ligase, so DNA ligase should be able to join the gene of interest with the plasmid, the open plasmid. So how do DNA ligase does this? By catalyzing phosphodiester bond between the gene of interest and the plasmid. So when they are joined together, they are and recombinant DNA is produced. The second modifying enzyme used is Taq polymerase, or commonly used is Taq polymerase. Taq polymerase is extracted from a bacteria. The difference between Taq polymerase and DNA ligase is Taq polymerase is heat stable, meaning they can withstand a high temperature. So usually Taq polymerase is used in polymerase chain reaction, because polymerase chain reaction is an application of genetic engineering that use a very high temperature. And Taq polymerase is purified from Thermus aquatus. That is why it is called Taq. T for Thermus aquaticus. This bacteria is found in hot spring bacteria. Tahu tak hot spring? Hot spring tu kolam air panas. Contoh kalau kat Sungai tu ada Sungai Kelah. Kalau dekat dengan Melaka ni ada Pedas, air panas Pedas dekat Negeri Sembilan. So Taq polymerase and DNA ligase has some similar function, but the difference is Taq polymerase can do its works in a high temperature environment. Thank you.